Hello, everyone. My name is Kyle Morin, and I am a third-year CMMB major at the University of Calgary. Today, I will be presenting my research project on characterizing autism risk alleles through behavioral analysis of mutated CHD8 and SHANK3B zebrafish. For a little bit of background, autism spectrum disorder, or ASD, is a neurological disorder affecting approximately 1 in 66 children in Canada. Symptoms of ASD include difficulty with social communication and interaction, restricted and/or repetitive behaviors, as well as delays in language, movement, and/or cognitive and learning skills. CHD8 and SHANK3 are syndromic autism genes within humans, referring to being a part of syndrome causing. CHD8 and SHANK3B are orthologs of those genes in our model organism zebrafish. Orthologs refers to the homologous genes that diverge due to speciation. There is little information on the behavioral progression throughout development within these genes. My objective for this project was to investigate how CHD8 and SHANK3B gene mutations affect locomotion and anxiety-like behaviors in developing zebrafish. My hypothesis for this investigation is that mutations in CHD8 and SHANK3B genes cause behavioral differences in zebrafish development, expecting to see changes in locomotive and anxiety-like behaviors. To investigate the effect of these genes mutations on locomotion and anxiety-like behavior, I first bred adult zebrafish containing the desired mutation in either CHD8 or SHANK3B genes. This resulted in a desired mutation being passed on to the offspring, allowing us to view development with the mutation. The larvae developed and were used in a behavioral analysis at 5, 7, and 10 days post-fertilization. The behavioral analysis consisted of larvae being separated into individual wells, with their movement recorded in response to alternating light and dark conditions as a startle experiment. At 10 days post-fertilization, the larvae were sacrificed and used in a polymerase chain reaction, or PCR, experiment, which basically amplifies the quantity of their DNA. Their amplified DNA was then put through an experiment called gel electrophoresis, which shows if the larvae contained a gene mutation or if it had no mutations present. Despite all the scary-looking graphs here, we can identify some results on the development of zebrafish larvae with either mutation of CHD8 or SHANK3B. Here, we can see CHD8 locomotive behavior results. As seen in the image on the left, larvae movements were tracked within the diameter of the circle. The top set of graphs shows the active speed of CHD8 mutants at 5, 7, and 10 days post-fertilization. Active speed being characterized by the larvae moving between 4 to 8 millimeters per second in response to the light startle. As we can see here, there were significant differences between the non-mutants in black and the mutants in blue at 5 and 7 days post-fertilization that disappeared at 10 days post-fertilization, suggesting the loss of a phenotype during development. The bottom set of graphs shows burst speed, which is characterized by movement greater than 8 millimeters per second. We can see here that the CHD8 mutant larvae began showing a burst speed phenotype as development progressed. Thigmotaxis is described as an anxiety-like behavior. When a zebrafish makes contact with the outer portion of the well for comfort, this is derived as an evolutionary adaptation to avoid predation. In the image on the left, we can see that thigmotaxis was scored as negative if the larvae were outside of the yellow circle and positive if within it. As we can see by the graphs, the CHD mutant larvae adopted a thigmotaxis or anxiety-like behavior as they developed. SHANK3B mutant behavioral analysis is still being conducted, but as we see here, there is no significant difference between the SHANK3B non-mutant in black and the mutant in red for active speed, burst speed, or thigmotaxis scores at 5 days post-fertilization. We are currently in the process of characterizing behaviors under the core umbrella of autism symptoms, specifically repetitive behaviors such as locomotion and anxiety-like in zebrafish. Thank you for listening to my presentation.